Hi Bio 111 students, and welcome to week 6. We're going to be discussing chapter 5 of the book, and chapter 5 is about cell membranes and their structure and functions. First off, we're going to talk about 4 major functions of the cell membrane. Number 1, it is a physical barrier to the environment outside of the cell. So you might say, well, why would it need a physical barrier? Well, that's because it needs some protection from the outside. And also it needs to maintain the shape of the cell so it can function. So that's number 1, the physical barrier. Number 2 is selective permeability. And if you think about the cell as being surrounded by a wall, we have certain gates in the wall that allow things in and out, and also the wall itself is permeable to certain things that the cell always needs. One of those things that is able to move in and out of the cell selectively are some of the ions and water. So there are certain channel proteins, and we'll talk about those later. But one of their important functions is to let the ions sodium and potassium in and out of the cell as needed. So, next important function of the cell membrane is endocytosis and phagocytosis. Or I should say exocytosis. We'll cover phagocytosis in a moment. It's a particular type of endocytosis. So endocytosis is when the cell membrane changes shape in order to take something into the cell. And exocytosis is when the membrane changes shape in order to expel something from the cell. And we'll talk about that in a few minutes too. Another important cell function is cell signaling. So there's a couple of ways that cell signaling works. One is communication with other cells. And another is IDing itself, identifying itself to other cells. And we'll get into how it does that, but just know that it is a very important function in that the body needs to be able to distinguish between self and non-self cells. Failing that, you end up with some serious problems, such as many of the cancers that the human body has to deal with, or infections that are able to circumvent our immune system. So those are the major functions, and now we're going to get a look at a basic drawing of a cell membrane. So before we draw a whole load of these, I want to show you one. Phospholipid is so named because it has a phosphate head and when you hear the word phosphate you should automatically be thinking PO4, a phosphorus and 4 oxygens. And that is highly polar. And it also has a non-polar part, and that is the two lipids that are attached. One of these lipids is saturated. The other one contains a double bonded carbon, causing this bend, and that makes it an unsaturated fatty acid. So you have a saturated and an unsaturated fatty acid. Those are the two lipids, and all together we call this a phospholipid. This part is polar, this part is non-polar. Words you've heard before in this class to describe polar and non-polar. Polar is hydrophilic. Meaning water-loving. Non-polar is hydrophobic. And you should know that right away because it is a lipid, and lipids are generally non-polar and they're generally hydrophobic. Hydrophobic means, when you think of phobia, we think of fears of something. So hydrophobic means afraid of water or not liking water. So that's one phospholipid, and now we're going to see what the cell membrane looks like. So one of the things they say about the cell membrane is that it has a phospholipid bilayer, and you'll see that they line up because of their polar and non-polar characteristics. So you might think we're going to see the heads of the next layer come in right there. You don't, because the next layer, you're going to first see the lipids of the next layer. And because this area here does not contain water, that is where all the hydrophobic parts of the phospholipid will be. So here we have our phospholipid bilayer, and that is the extracellular area. So we'll call this ECF, extracellular fluid, and this is intracellular fluid, or the cytoplasm. So what we have here is inside the cell and outside the cell, and we have between them we have this barrier of a phospholipid bilayer, called a bilayer because there are two layers of phospholipids. Within this bilayer, we are going to see some other things, and one of those things are membrane proteins. We have two types of membrane proteins. We'll draw our proteins in red so you can see that they're different than the phospholipids. This protein is called an integral protein. The reason why it's called an integral protein is because it passes through the top and bottom layer of the phospholipid bilayer. So it is integral to the membrane, the entire membrane. The next protein we're going to talk about is a peripheral protein. This is a protein that resides on either the inner or the outer layer of the cell membrane. So we could have one there, and we could also have one here. And sometimes those peripheral proteins are glycoproteins or something like that, but we'll talk about that later. They have different functions. So we were talking about selective permeability, and that basically means that only certain molecules can pass through. So we'll say selective permeability. Let's say we have a potassium molecule. Let's use a different color. And this protein allows the potassium model to pass through or pumps it through, which if you've heard of the term sodium potassium pump, well, that in cells in the cardiac cells in the heart, they have sodium potassium pumping proteins that basically constantly are pumping potassium into the heart in order to allow the electrochemical reactions that are necessary for our heart to beat happen. So we have the phospholipids. Another thing we might have is membrane cholesterol, and we'll find that between the phospholipids. So here we have a membrane cholesterol. Maybe we'll put another one here. And again, I'm using different colors so that you guys will see that they're different. We have some of the proteins that are peripheral proteins. They might work as recognition proteins. A recognition protein identifies this cell to other cells that come by, and also it recognizes foreign cells. And this becomes very important when you're talking about immune system cells, such as the white blood cells of our body. Some of them are recognition. Their job is to recognize and tag, and others they have to recognize and kill by engulfing foreign material or foreign cells to our body. So another type of protein besides recognition protein is, let's go with blue. Sugar, we'll color that blue today. And these are glycoproteins. They are a specific type of peripheral protein. And so glycoproteins are proteins with some carbohydrate attached. And we call that a glycoprotein because it has a glyco, which is a carbohydrate attached. We have transport proteins. This could be one of them that allow for to selectively take things that the cell needs in through this gateway here into the cell. So stuff that's out here would be taken in selectively. We have receptor proteins. The receptor proteins' job is hormone responses. One more thing I want to say about glycoproteins, and I thought it was just very interesting, that let's say this is a white blood cell, and we have a glycoprotein on a T cell. A T cell is also known as a killer T cell. That is the type of white blood cell whose job it is to go and actually kill foreign virus or cellular material by engulfing it, and digestive enzymes take care of the rest. With HIV, you end up with a problem on this particular this particular carbon on the glycoprotein, and it's called the CD4. And that's all it takes for HIV to get by all of those killer T cells and to inactivate them. And the reason why HIV is called a human immunodeficiency virus is because by taking out all of those T cells, it makes them useless. And without T cells, something else is going to come along and get you. So some sort of opportunistic infection, whatever it is, often it's pneumonia, can get you if HIV is untreated inside you. We also have some enzymes in the phospholipid bilayer, and we have connection proteins. We saw that for when we were talking about the proteins that connect two cells together in the last section. So let's talk about the concentration gradient. So with any gradient, it's like a hill. And stuff generally flows downhill with no energy needed for it to happen. So that's the general direction of movement. So things flow from a high concentration to a low concentration. And no energy is needed to get that done. That is the definition of a concentration gradient. We're going to talk also about diffusion. Diffusion and the concentration gradient go together because we will generally have stuff go from a high concentration to a low concentration. And an example that I would use of diffusion is take a beaker, and let's say this is a beaker of water, and so we see it's clearer, and then we take a drop of red dye and we put it into the beaker right here in the middle. Well, right here the dye is highly concentrated and there is none or less of it out here, and slowly but surely this entire thing will become pink. This is called diffusion. Now, there are some factors that definitely affect the speed of diffusion, and one of those is molecule size. So if these molecules are smaller, they will go faster. So molecule size affects diffusion. The next thing that any of you that drink tea will know this right away. Temperature affects diffusion. So when you're making your cup of tea, you'll notice that it's much more easy to make a cup of tea with hotter water than to try and make it with colder water. Another thing that affects diffusion, the concentration gradient itself, basically how steep it is. So in this case, it is quite steep because you have 0 here and you have a lot here. In certain cases, the concentration gradient will be less steep because there's already some of that dye in there. So the concentration gradient affects diffusion. So if the concentration gradient is less steep, this you're going to see fast diffusion, this you're going to see slower diffusion. Now, I want to talk about diffusion. There is no membrane involved in diffusion, and I want to talk about osmosis. Osmosis differs from diffusion because there is a membrane involved. So here we have our water, and here we have our dye, but this time our dye is in a semi-permeable bag, which is sort of what a cell would be. And let's say attached to our semi-permeable bag we have a pipette or something like that. We would see water movement from the water into the bag, the semi-permeable bag, because the water will want to move to an area of higher concentration. Now, there is a big difference between osmosis and diffusion, and that is there is a semi-permeable membrane involved, and also the other difference is water is the only thing that moves in osmosis. So I would definitely put in osmosis, water only, high cons, and it's water only to the higher concentrated area. And so people do mix up diffusion and osmosis, but as you can see, there's not much reason to do it. There are major differences. Another beaker. This time I put in water, and I sprinkle in some salt. And so the salt dissolves in the water. Here's our salt cellar. So the salt dissolves in the water, and we call the water the solvent. And we call this stuff the solute. So whatever it is that's dissolved is called the solute, and the stuff that dissolves it is called the solvent. So a way to think about it is solve is part of the word dissolve. So the stuff that's dissolved, the stuff that does the dissolving is called a solvent. And the solvent that we have most of in the world is H2O water. So now we're going to talk about passive transport and energy requiring transport. So passive transport usually goes this way down the concentration gradient. Active transport is usually, or energy requiring transport is usually going the opposite direction. Energy required. You could think of it as letting a car roll down a hill or trying to push a car up a hill. Which one requires the energy and which one doesn't. So we have passive transport and energy required transport. Simple diffusion is what we talked about earlier. Facilitated diffusion is when something needs to get involved in order to help the diffusion to happen. So facilitated diffusion is often what's needed for certain molecules that are repelled by the cell membrane. So like we talked about earlier, simple diffusion is no energy, simply just passing on through, no problem. Diffusion, passing, from a high concentration area to a low concentration area, that's simple diffusion. Facilitated diffusion, no energy is required, but sometimes we need to overcome the repulsive forces of the cell membrane itself, and we use facilitated diffusion to do that. And this often happens with polar solutes. And we were talking about one of those earlier, K+. Potassium is one of those polar solvents. Actually K+ is not, I'm sorry to say. So it's just any polar molecule needs polar solute, needs some facilitated diffusion to get into the cell. Active transport is another type. Active transport is how K+ potassium gets into our cells, and it's against the gradient. Facilitated diffusion is with gradient, but the polar solute is would be repulsed by the cell membrane if there wasn't a protein there to help pull it in, and that is facilitated diffusion. Active transport is when you have high or low to high. So this is the pushing the car up the hill. Low to high concentration movement. And this is the case when you are pushing potassium molecules into nerve cells. That already have more, and that's the thing about nerve cells, they already have more than the extracellular fluid, but we still need more of them inside the nerve cells in order to do their functions. So that's the reason why we need some active transport. Active transport requires energy, and as we know, energy is measured in ATP. So now let's talk about phagocytosis and pinocytosis. So, okay, we have a white blood cell, and these are the ones that have the nucleus. If you guys remember, looked a little bit like this. And this white blood cell that has a nucleus comes across something it doesn't like to see too much, a little virus or some other foreign material. And what happens is the cell membrane changes shape. Think about this like Pac-Man almost. And engulfs this guy. And so what you end up with is, this is 1, 2, we end up with is this with a little vesicle inside it with our little undesirable thing in there. And inside the vesicle it will be destroyed. Lysosomes will attach and destroy by using enzymes, destroy the foreign object and thus eliminate the threat. So that's phagocytosis. Pinocytosis, spelled this way, and also I should give you the spelling of phagocytosis. It's over here. What I'm going to show you now is pinocytosis, and that is when the cell needs fluid, and as a result we might see something like this develop in the cell. It'll start off as a depression on the cell membrane, and then we will see this happen where it takes some extracellular fluid and finally engulfs it. And once it's satisfied that there's nothing dangerous in there, it will become part of the extracellular fluid or intracellular fluid. So let's talk about carrier proteins. Carrier proteins. So let's say it's one of these guys, and he's in the cell wall, and the carrier protein binds to something foreign, something the cell needs, and so it's a chemical bond that's formed, and then once it has identified that this is what the cell needs, it will pull in a couple of steps this needed object or needed substance will be pulled inside the cellular membrane. And then we have channel proteins, which are somewhat similar but more passive in that channel proteins, they're hydrophilic in parts, and that allows passage of some of those polar things we were talking about. So our channel protein is basically like what I drew earlier. It's a large protein with a channel in the center. If you can imagine, this is a channel, and I'm just showing a cutaway because it's actually from the top it would look like this. And the channel protein has a channel in the center, and that allows the passage of things that the cell needs. And then we have aquaporins. Aquaporins are interesting. They are a sort of channel protein, but they are always gathering water, hence the word aquaporin. Aqua for water and pour for the pore that's always in the center and it's open, much like a channel protein, but it functions to get water in quickly, and so aquaporins are known for their high rate of water intake. So now let's talk about hypotonic, isotonic, and hypertonic. So we have 3 solutions. Let's say they're salt solutions. Okay, one is hypotonic. One is isotonic. And the last one is hypertonic. Now, tonicity actually refers to concentration. So, hypotonic would be, and its tonicity means in relation to the ordinary concentration in the human body. So let's say the ordinary concentration in our cells. If we're talking about humans, of course we could be talking about any animal or plant, but in our case, normal isotonic is 0.9% sodium chloride. So that's why when you're in the hospital, you'll see a lot of 0.9 sodium chloride, which is 0.9 saline. Normal saline is what you'll see being given to patients that need some fluid, because it's got the same tonicity as our blood cells. So when you put, let's say you put a red blood cell into this fluid, we'll see no change. So there's really no net movement of water either direction. Now let's say we put the same cell into hypertonic, which could be, quite salty. Let's say it's 4%. So that's, 4 times this 4% NaCl, so 4% salt. What we'll see is the water from inside the cell will actually travel out, and the cell will shrink. And in some cases they call it crenating, but it will shrink and it will die if this process is just left happen. If more water isn't introduced to dilute this, we'll see this harm come to this blood cell, and that's what we see with dehydration inside the human body. Next, we'll talk about hypotonic, and what we'll see is the cell will swell and swell and swell until eventually it just bursts. And that's called lysing or bursting of the cell. So that's the effect on living cells of those type of solutions. In the case of plants, what we'll see is something similar, but we'll see cell walls involved. So you'll see the cell just shrink, and in this one the cell will be unaffected, and in this one we'll see the cell, it can't quite burst, but what you'll see is the vacuole will be bigger and bigger and bigger, and finally it'll be so pushing the rest of the contents so tightly against the walls it won't be a functional cell. So we're going to talk about exocytosis. We talked about pinocytosis earlier, and now you'll see exocytosis. So with exocytosis, here's our cell membrane. And here's a vesicle with stuff in it that we don't want approaching the cell membrane. And once it gets there, we'll see it attached to the inside of the cell membrane, and an opening will form, and then this stuff will go out into the ECF, the extracellular fluid, and this is called exocytosis. So we have an arrow going from this, and finally it will become just another part of the cell wall. This whole part here will just become part of the cell wall, so that it goes this way. As we saw with pinocytosis, all you're really doing is changing the arrows directions, because they're taking in stuff that we need. Let's say that stuff is fluid. So pinocytosis, also called the cell drinking or hydration of the cell, and again it will be the taking in of fluid. So that's all we have for you today, and thank you very much.